session are two plenary keynote lectures which exemplify the idea of this year's conference, so bringing basic science and medicine together. And the two lecturers who will be presenting their talks show us how they tackle this problem of bringing the basic science discoveries pertaining to fundamental things of such as cell cycle or DNA repair, and how those can be converted into applicable treatments for human suffering from cancer. So our first lecturer is Professor Peter Siciński, who is now working in Boston in the Dana-Farber Cancer Institute. He's running his own department of genetics at the Harvard Medical School, and he originated from Poland, from Warsaw Medical University, if I recall correctly. He traveled to Cambridge and then found his place in Boston, initially at MIT at Dr. Weinberg's lab. And now he's leading his research group focusing on cell cycle machinery and utilizing those as therapeutic targets in cancer. So his talk will concern specifically the cyclins and novel drugs and all the exciting things that are happening right now in the field. And then we'll have the second lecture with questions between one and the other. So thank you, Peter, for coming, and the floor is yours.
I would like to thank Wojciech for inviting me here. It's really a great pleasure and honor. Can you hear me? It's a great pleasure and honor to come here and to tell you about our work. And actually, Wojciech instructed me. No. Okay. Is it close enough, or should I put it into my mouth? Then I can. Okay. I don't know. I can hear myself very well. Is it okay in the back? Okay, so I was instructed not just to talk about the latest research, but rather to give you some overview about our past work, and then continue with the most recent work. So broadly speaking, our lab is interested in understanding what are the mechanisms. Oh, that's enough, thank you. What are the molecular mechanisms that govern cell division? How those mechanisms are different in normal cells and in cancer cells, and how we can exploit those differences for cancer therapy. So both in normal cells and in cancer cells, the cell division is driven by the core cell cycle machinery operating in cell nucleus. The core cell cycle machinery is composed of multiple proteins called cyclins, and these are represented here by pink ovals. And one of the major functions is to bind and activate the kinase partners called cyclin-dependent kinases, or CDKs. And those different cyclin-CDK complexes phosphorylate cellular proteins, thereby driving cell cycle progression. Now, as you can see from this slide, there are several different cyclins and cyclin-dependent kinases. For the purpose of this talk, I will focus on three D-type cyclins. These are three different proteins, cyclin D1, D2, and D3. Each of them can activate either CDK4 or CDK6. Indeed, most of the cancer field is most interested in cyclins and cyclin-dependent kinases operating during this phase of the cell cycle, G1 phase of the cell cycle, because all these proteins are hyperactivated in virtually all human tumors. The cyclin D1 itself is the second most frequently amplified gene across all human cancer types. Its kinase partner, cyclin-dependent kinase 4, or CDK4, is ranked as number 4. Now, in particular, this is just gene amplification, but of course you can get hyperactivation of a protein through different mechanisms. And for example, at the protein level, cyclin D1 is overexpressed in over 50% of breast cancers, as illustrated on this slide, showing us a section through a breast cancer with the staining for cyclin D1 in tumor cells. So when those proteins were discovered in late 1990s and the previous century, the dogma in the field was that each cyclin and each cyclin-dependent kinase is required for cell proliferation. So although those proteins seem like a potentially good therapeutic targets, this dogma precluded targeting them for cancer treatment. But our lab, and independently beautiful work from our competitors from Madrid, Mariana Barbacid and Marcos Malumbres, has shown that individual cyclin proteins are actually not needed for proliferation of normal cells through enormous redundancy. And Marcos and Mariana have shown that you can remove the individual cyclin-dependent kinases, and yet the cells in the mice develop essentially normally. So this lack of requirement, in this case for cyclin D1, in normal development, together with its so well-established role in breast cancer, prompted a former student in the lab, Chuan-Yin Yu, to ask if oncogenic signals through cyclin D1 in breast cancer setting, what would happen to if cyclin D1 was removed? Would those signals be able to drive tumor formation? And experimentally, she took cyclin D1 knockout mice and crossed them with 4 strains of breast cancer-prone mice. These mice developed breast cancers because due to targeted overexpression of particular oncogenes, which are listed here in the mammary epithelium. And she generated transgenic mice with those oncogenes in the breast at the cyclin D1 wild-type background, which we know will develop tumors, and in cyclin D1 knockout. And this is our experimental group. Now, Chuan-Yin is presenting the data as a percentage of tumor-free mice. Of course, at the beginning, all mice are tumor-free, and eventually they all develop tumors. These are the tumors driven by Wnt1 oncogene. In black, cyclin D1 wild-type background. In red, cyclin D1 knockout. And there is no major difference between those curves. Likewise, in case of MYC-driven cancers, cancers driven by the MYC oncogene, there was no difference in occurrence between cyclin D1 wild-type and cyclin D1 knockout. But the dramatic different picture emerged when Chuan-Yin looked at mice in which the cancer was driven by this cancer-causing gene HER2. In this case, the control mice developed tumors, whereas cyclin D1 knockout mice were totally protected against tumors. They never developed any tumors. Now, HER2 signals to the cell cycle machinery through the RAS oncogene, and you actually, more through luck rather than through some intelligent design, also included this strain of mice, which overexpress RAS oncogene in the mammary glands. And it was very gratifying to see that also these mice were protected against breast cancer in cyclin D1 knockout background. This is cyclin D1 wild-type background. So Chuan-Yin concluded is that cyclin D1 is not required for breast cancers driven by MYC and Wnt1 oncogenes, but is essential for HER2 and RAS-driven tumors. Now, cyclin D1 activates CDK4 and CDK6. In fact, in the mammary glands, it's CDK4. What I didn't tell you in the introduction is that cyclin D1 performs several CDK-independent functions that actually our lab is also actively studying. Now, Chuan-Yin wanted to test whether the interaction between cyclin D1 and CDK4 is responsible for this, for breast cancer initiation, and therefore she took knockout mice lacking kinase partners of cyclin D1, CDK4, crossed them again with this transgenic strain, and generated either CDK wild-type control group or CDK knockout, the experimental group. And exactly as she found in case of cyclin D1 knockout mice, she found that mice lacking CDK4, catalytic partner of cyclin D1, were also totally protected against breast cancers. These are the control mice that they develop tumors as expected. So we can remove cyclin D1, or we can remove CDK4, and we can make mice resistant to breast cancers. But when you start disrupting the complex, several secondary things happen. For therapeutic purpose, we wanted not to knock out those proteins by silence the kinase activity of this complex. And to do this, we took advantage of a cyclin D1 point mutant generated many years ago by Phil Hines. This is a point mutant of cyclin D1, which can bind CDK4 and CDK6, but it's totally unable to activate its kinase activity. So Mark Landis, Mark was actually a student in Phil's lab, but came to my lab to do this work. He made a knock-in mouse when he inserted a point mutation into cyclin D1. So he changed a single base pair in the mouse genome, which made cyclin D1 kinase dead. So he made mice which express cyclin D1, express CDK4, cyclin D1 binds CDK4, but the complex is catalytically inactive. These mice develop normally, but when crossed with this strain of mice which triggers breast development, were again completely resistant to breast cancers. So I think it's actually quite amazing that the mouse has 3 times to the 9th base pairs, and then you can choose a single base pair in the mouse genome, and you can make mouse totally resistant to breast cancer. Of course, we need to know which base pair to mutate. So those results led us to conclude that indeed cyclin D1 CDK4 kinase is required for the initiation of tumors. But of course, for therapeutic purpose, we need to know whether cyclin D1 is needed for progression of breast tumors. To address this question, another postdoc in the lab, Yoon Choi, generated conditional cyclin D1 knockout mice. And when crossed with mice which ubiquitously express tamoxifen-inducible Cre, we can develop mice with cyclin D1, and then when they come to the lab, give mice tamoxifen, delete cyclin D1 everywhere, and observe mice for 1 year. And as we found for cyclin D1 knockout mice, also acute shutdown of cyclin D1 had no obvious phenotypic consequences. So now, Yoon took her conditional knockout mice when she can turn off cyclin D1 anytime and crossed them with breast cancer-prone mice. She waited until the tumors developed tumors in the presence of cyclin D1, and then she switched off cyclin D1 in the whole mouse, including in the tumor. Once again, there was no consequence for the mouse physiology. However, the tumors arrested their growth. This is shown here, the size of the tumors treated with, in mice treated with vehicle only, or with, this is the mice in which we did not knock out cyclin D1, and this is in cyclin D1 deleted background, and this is the quantification. Now, at the same time, one pharmaceutical company, Pfizer, developed CDK4 and CDK6 inhibitor called palbociclib. And in the preclinical study, Yoon tested how well this inhibitor works by giving these inhibitors to mice that develop breast cancers. So once again, there was no major effect on mouse physiology, but inhibition of CDK4/6 kinase virtually phenocopied the cyclin D1 knockout. That is to say, it arrested cell proliferation and it blocked tumor growth. So we concluded the cyclin D1 CDK4 kinase is required for breast cancer maintenance, but not for normal mouse physiology. And based on this work, and also again on really great work from our competitors, from Mariano Barbacid in Madrid in Spain, now more pharmaceutical companies developed CDK4/6 inhibitors, in addition to Pfizer, Novartis, and Eli Lilly developed those inhibitors. The first inhibitor that was tested in clinical trials was the Pfizer compound, palbociclib, which was treated for patients with estrogen receptor positive breast cancer, metastatic breast cancer. And the patients are either treated with standard therapy, shown here in red, which is anti-estrogenic therapy, or standard therapy, anti-estrogenic therapy plus palbociclib. And as you can see, this led to basically the doubling of the progression-free survival time. So based on this, this compound received breakthrough therapy designation status from our Food and Drug Administration. And then in next years, also abemaciclib and ribociclib, the other two inhibitors, received this breakthrough therapy status. They have been now approved for treatment of breast cancers. And currently, there are about 200 clinical trials worldwide with CDK4/6 inhibitors, both for breast cancer, but also for all different cancer types. So in the last years, there have been enormous amount of studies which has shown that the inhibition of these proteins in cancer cells blocks cancer progression, not only in breast cancers, but also in several other tumor types. And this is why those 240 clinical trials are not restricted to breast cancers. However, we have found, and I should add that not only we, but also independently, Yannis Zaifanti's lab at NYU have found exception to the rule. When we shut down cyclin D1 in mice, in mice which had leukemias, we observed not only cell cycle arrest, but also eradication of tumors. That is to say, the tumor death. This is actually a quantification of the tumor. Here we are shutting off using conditional knockout cyclin D3, because in leukemias they mostly overexpress cyclin D3. So when we turn off cyclin D3, the tumor cells disappear, as you can see at the quantification here, and they undergo apoptosis, as shown here by the by annexin staining. We can recapitulate the same not by knocking out cyclin D3, but rather by treating mice with the inhibitor, tumor-bearing mice with the inhibitor of CDK4, CDK6, palbociclib. And as you can see, the tumor cells disappear, and the apoptotic rate goes up. So we concluded that cyclin D3, together with CDK4/6, plays a pro-survival function in human leukemic cells, but the mechanism was not known. So Haijian Wang, who is still a postdoc in the lab, although she's currently on the job market, spent 5 years trying to understand this molecular mechanism of this post-survival function, hoping that by understanding what happens in leukemias, we can make palbociclib more potent, maybe also for other tumor types. So first of all, the first thing she noticed is that the human leukemias, of all those different combinations of cyclin D, CDK4 combinations, I mentioned that you have cyclin D1, D2, D3, each of them can bind CDK4 or CDK6. The leukemic cells express only CDK6 and only cyclin D3. So now we asked what are the substrates. So we concluded that cyclin D3 CDK6 is driving this pro-survival function through phosphorylation of some proteins in leukemic cells. To identify those proteins, we ask what are the binding partners of this kinase in leukemic cells, because quite often kinase physically binds the substrates. And when we look at the substrates identified by mass spectrometry and the gene ontology analysis, we did not find cell cycle as an enriched function. What we found is glycolysis and the functions, all of them related to glucose metabolism. In particular, looking at those interactors, even without gene ontology analysis, Haijian immediately noticed the presence of two rate-limiting enzymes in the glycolysis. 6-phosphofructokinase, which is called PFK1. Sometimes I'll call it PFKP, not to confuse you, but PFKP is the major isoform of this enzyme that is present in leukemic cells, and pyruvate kinase M2. Those two enzymes catalyze the irreversible steps in glycolysis. So we identify those proteins as a binding partners, but of course we wanted to find whether we are looking for substrates. So we were very happy to see that indeed, when we immunoprecipitate CDK6, shown here from two different leukemic cell lines, and perform kinase reactions, CDK6 can phosphorylate both PFKP and PKM2. Importantly, when we add the palbociclib to the reaction, the inhibitor of CDK4, CDK6, we can extinguish this reaction, indicating that this is a specific phosphorylation. A surprise came, and I say a surprise because the current thinking is that cyclin D1, D2, D3 together with CDK4, CDK6 are tissue-specific isoforms of the same enzyme. But here the surprise came when Haijian did kind of a control experiment and tried other combination of cyclin D, CDK4, and she found that only cyclin D3, CDK6 of all those different combinations can phosphorylate the metabolic enzymes. The cyclin D1, CDK6 cannot do it, cyclin D1, CDK4 cannot do it, only cyclin D3, CDK6 can phosphorylate those enzymes. So now we found that the phosphorylation of endogenous PFKP and PKM2, the cyclin D3, CDK6, indeed phosphorylates the endogenous enzymes, because when we inhibit cyclin D3, CDK6 by using palbociclib, then the phosphorylation of the endogenous PFKP and PKM2 goes down, as you can see here. Now, through a series of biochemical experiments, we found that, meaning Haijian has found, that actually cyclin D3, CDK6, by phosphorylating those two enzymes, it inhibits the activity. So here in this experiment, in cells, she's either expressing the empty vector or cyclin D3, CDK6. And as you can see, cyclin D3, CDK6 overexpression reduces the activity of endogenous PFKP and activity of endogenous PKM2. Importantly, when we overexpress cyclin D3 together with kinase dead CDK6, now there is no effect, kind of confirming that this is a kinase-dependent function. So whereas the overexpression of cyclin D3, CDK6 lowers the activity of those enzymes, inhibition of cyclin D3, CDK6 by treating cells with palbociclib activates those enzymes, as shown here. These are three different leukemic cell lines. This is before, and this is after treatment with palbociclib. So we were very excited by those findings because it's known from cancer metabolism that cancer cells like to inhibit those two enzymes. The rationale for this is by inhibiting those two enzymes and by inhibiting the glycolytic flow, the cancer cells promote the carbon flow into pentose phosphate pathway and into serine pathway. These are very important pathways because the pentose phosphate pathway generates NADPH, which is the major factor countering increased levels of reactive oxygen species, which happen in tumor cells. And also the second pathway promotes production of NADPH through one-carbon metabolism, and also it promotes production of GSH. So by this way, the cancer cells shunt the carbon into those pathways, therefore countering high reactive oxygen species levels. Now, our little contribution to this field was showing that in cells which express this isoform cyclin D3-CDK6, it is cyclin D3-CDK6 that inhibits those enzymes. So from this, we can make some predictions. If this model is correct, then if we add inhibitor of CDK4-CDK6, we are going to de-inhibit PFKP, there will be more carbon going into the glycolysis, and therefore less pentose phosphate flow and less second pathway flow. So we can actually quantify by taking the cells, giving them radioactively labeled glucose, and then following the flow of the labeled carbon by mass spectrometry. So indeed, as we predict or expect from this model, if we treat cells with the CDK4/6 inhibitor, the flow of carbon into pentose phosphate pathway and second pathway goes down. We can predict from this model that the decrease of the flow into PPP and second pathway will lead to decrease of NADPH, decrease of GSH, and increase of reactive oxygen species levels. And that's exactly the case. So these are 4 different cell lines. Before and after treatment with palbociclib, the NADPH levels goes down, the GSH goes down, the reactive oxygen species goes up, and these cells now undergo massive apoptosis. So in summary, this work kind of is consistent with the model, is that cyclin D3-CDK6 regulates the carbon flow into pentose phosphate pathway and serine pathway, and thereby promotes the production of NADPH and GSH by inhibiting 2 glycolytic enzymes. And then by inhibiting CDK4/CDK6, we can again rewire cancer cell metabolism in a way that is going to trigger tumor cell killing. But this was all done in leukemic cells, and we wanted to know whether this happens also in other tumor types. Leukemic cells are unique because they express high level of cyclin D3-CDK6. Other cancer types have either combinations of cyclin D1, for example, CDK4. But we wanted to know maybe there is a subset of normal tumors that happen to express high level of cyclin D3-CDK6. And to test this prediction, Haijen took the Human Cell Encyclopedia, which is like a compendium. This is the list of 1,050 cell lines, and she ranked them from the top to the bottom for the level of CDK6 and for cyclin D3. And she picked top 20 cell lines. So these are top 20 cell lines that uniquely express high level of cyclin D3-CDK6 RNA. Of course, she verified the protein level, and then she treated these cyclin D3-CDK6 high cell lines, non-leukemic. These are non-leukemic cell lines. These are from solid tumors with the palbociclib. And except for 2 cell lines, except for this one and for this one, 18 out of 12 underwent indeed apoptosis when CDK4/6 was inhibited, as shown here. This is before and this is after inhibition. This is before and this is after inhibition of CDK4 and CDK6. Like in leukemic cells, also in these non-leukemic cells, but expressing high level of cyclin D3-CDK6, upon treatment with palbociclib, the phosphorylation of endogenous PFKP and PKM2 went down. Like in leukemic cells, the inhibition of CDK4/CDK6 increased the activity of PFKP and PKM2 in tumor cells. And like leukemic cells, this led to the reduction of NADPH and GSH levels, increased level of reactive oxygen species, and triggered tumor cell apoptosis. So lastly, we wanted to know whether this phenomenon, which we are seeing in cell line, could actually operate in real tumors. And the closest you can get to real tumors are the patient-derived xenografts. We actually collaborated with Dr. Hui Gao from Novartis, and what they did is they took 33 primary melanomas and implanted into the mice, and either leave the mice untreated or treat them with ribociclib. Ribociclib, like palbociclib, inhibits CDK4/CDK6, but since this is collaboration with Novartis, they are using their own compound. But it works the same way chemically by inhibiting CDK4/CDK6. So here you can see the growth pattern. So each color corresponds to different tumor. There should be 33 in total. If untreated, if mice are untreated, those tumors grow quite aggressively. if we treat the mice with the ribociclib, CDK4/6 inhibitors, some tumors don't care, they continue growing. Some tumors most slow down their growth, and in a few cases, they actually stop growing, indicating like nearly complete cell cycle arrest. But there were 3 unusual tumors, 3 out of 33, which behaved in an abnormal way, namely, they shrunk. They underwent apoptosis, and either in the case of this tumor, almost all cells were eradicated, although eventually there was some resistance, and the tumor regrew after 250 days. In case of this tumor was eradicated permanently, and in this one the few residual kind of small tumor after almost 1 year. So we got those tumor samples in a coded way, and we tested the level of cyclin D3 CDK6. And the results are shown here. This is the Western blotting for CDK6 and for cyclin D3. In the first lane, you see a positive control, the leukemic cells, which, as I told you, have high level of CDK6 and cyclin D3. And this 1, 2, 3, these are the tumors which underwent regression, which underwent apoptosis. These cells express high level of cyclin D3, CDK6, like leukemias. And this is a selection out of 30 tumors which did not undergo apoptosis, and they express relatively low level of cyclin D3, CDK6. So now we took those 3 tumors, those cyclin D3 CDK6 high tumors that underwent regression, and 3 randomly selected tumors from here, the ones that have low level of cyclin D3 CDK6 did not undergo regression. We put them back into the mice, but now we are not doing any long-term experiment, but rather we want to confirm short-term what happens if we give mice injection of ribociclib. So we inject tumor-bearing mice with inhibitor, then we collect tumor, and we look at the phosphorylation of PFKP and PKM2. So in cyclin D3 CDK6 high tumors, administration of an inhibitor reduced the phosphorylation of these enzymes, both PFKP, PFKM2. This is first, second, the third regressing tumor, but there was no effect, as one would predict, on the cyclin D3 CDK6 low tumors. In the regressing tumors, we saw upregulation of activity of PFKP and PKM2, like in cell lines, but there was no effect on those parameters in cyclin D3 CDK6 low tumors. Likewise, the GSH went down, and reactive oxygen species went up in these tumors, but not in low tumors, and these tumors underwent apoptosis. These did not. So this is the summary for this part of my talk, is that cyclin D3 CDK6 kinase plays a pro-survival function in human leukemic cells by phosphorylating 2 rate-limiting glycolytic enzymes, PFK and PPKM2. This function is independent of the textbook role of cyclin D-CDK4 in phosphorylating retinoblastoma protein. I didn't show you the data, but of course I'm very happy to show it later. And the review proposed, although with the caveat that this is a very small sample of 33 tumors, that measuring cyclin D3 CDK6 levels may allow one to identify tumor subsets that undergo apoptosis upon CDK4/6 inhibition. So I told you until now, hopefully, that in addition to driving cell cycle progression, those cyclin CDK complexes perform cell cycle independent functions. In case of cyclin D3 CDK6, this is driving or rewiring tumor cell metabolism. We actually found recently another function, cell cycle independent function of these complexes, not for cyclin D3 CDK6, but cyclin D-CDK4 is the role of cyclin D-CDK4 in regulating anti-tumor immune surveillance. I should add that this story is an equal joint collaboration with Wayne Way Lab at Beth Israel Medical Deaconess and is Gordon Freeman in our institute. So I'm putting this slide since I'm not an immunologist and know very little about immunology, just to remind myself is that the cytotoxic T cells recognize the antigens, the tumor cell antigens. However, they are kept in a silenced or exhausted state because the tumor cells often upregulate the inhibitory protein called PDL1, which binds PD-1 receptor on T cells and silences them and exhausts them. And one of the major breakthroughs in cancer therapy in recent years is the so-called checkpoint therapy. And this is, for example, shown here. For example, patients can be treated with anti-PD-L1 or anti-PD-1 antibodies that would disrupt this inhibitory interaction, therefore unleashing the killing of tumor cells by the cytotoxic T cells. Fortunately, this therapy works in a small, it works like a charm. And I show you in the next few slides, it also works in mice. But unfortunately, it works in a very small subset of patients. And there's data indicating that actually the level of PD-L1, the high level of PD-L1 on tumor cells correlates with very good response to this immunotherapy. So there is a great interest to understand what are the mechanisms that are controlling the level of PD-L1 in tumor cells. So working with Gordon Freeman and Wen Wei, we found that cyclin D CDK4 negatively regulates PD-L1 protein levels. This is our favorite genetic system in fibroblasts. These are the wild type, and this is triple knockout fibroblasts, probably generated by Professor Chemerich many years ago. And as you can see here, there is a dramatic upregulation of PD-L1 levels in these fibroblasts. Of course, you could say these are the fibroblasts that are passaged without cyclin D1, D2, D3, so maybe something strange happened. But this is a acute shutdown of D1, D2, D3 using conditional mice that Yun-Chon made. Again, there is upregulation of PD-L1. This upregulation is at protein level because RNA, if anything, goes down. We see upregulation of PD-L1 also in a knockout of CDK4. And we can also increase PD-L1 levels by knocking down cyclin D1 or cyclin D3 in human cells. So this is not mouse-specific effect. Now, of course, we don't want to study PD-L1 in fibroblasts, so we immediately switch to tumors. We take MMTV Wind white mice, which I showed you in one of the first slides, cross them with either wild type or cyclin D1 knockout mice, and then collect breast tumors, which are either cyclin D1 wild type background or cyclin D1 knockout background, and then look at, so each number corresponds to different tumor from different mouse, and you look at the level of PD-L1. And again, when you knock out cyclin D1, the PD-L1 level goes up actually in the tumor. This is MMT, the mixed strain, and again, this is cyclin D1 wild type background, cyclin D1 knockout background, and the level goes up. Also, we can upregulate the levels of PD-L1 not by knocking out cyclin D1, by inhibiting the kinase activity. So here are human cancer cell lines, not and treated with a palbociclib. This is the level of PD-L1, and in several of different cell lines, those levels of PD-L1 are upregulated. We found the same in mice. So here we are taking MMTV HER2 mice. We give them palbociclib the same way that Yoon Choi did it 6 years ago. But now we give mice palbociclib, and we stain the tumors for PD-L1. So this is immunostaining. This is a vehicle-treated tumor, and this is tumor from mouse that receives palbociclib. And then indeed the level of PD-L1 goes up. This is actually accompanied by the decreased numbers of the tumor-infiltrating T cells or CD3 positive cells. This is the quantification. This is tumor infiltrating cells in control and in palbociclib treated mice. We can do the same, yet another system. This is an allograft model, mouse model. When you take mouse colon cancer cells, we put them into the mice, and the mice develop tumors. We either treat them with vehicle or with palbociclib, and we see upregulation of PD-L1 shown by Western blotting, upregulation of PD-L1 shown here independently by FACS analysis, and the decrease in the number of tumor-infiltrating lymphocytes. This is the slide that I don't want to take credit for because this is, I'm just using this slide to show that cyclin D1 CDK4 controls the level of PD-L1 indirectly. But this exact mechanism was worked by Wen Wei's lab, and our little contribution, so what Wen has found is that PD-L1 is actually degraded through the ubiquitin ligase, which is an F-box protein, which binds the substrate, uses protein called SPOP. And our little contribution, Hygen's little contribution, was to find that cyclin D1 CDK4 actually phosphorylates SPOP. And then when he worked out the rest, namely when cyclin D1 CDK4 phosphorylates SPOPs, it stabilizes SPOP. SPOP is now there to degrade PD-L1, so we have low level of PD-L1, and then when we inhibit CDK4, CDK6, the SPOP becomes destabilized, and therefore the PD-L1 level goes up. regardless of this mechanism, we were really excited about this finding because I told you at the beginning is that a high level of PD-L1 predict good response to anti-checkpoint therapy. So we were speculating that maybe combining the immune therapy together with CDK4/6 inhibitors could increase the efficacy of anti-checkpoint therapy, of immune therapy. And to test this, we took mice and either implant the colon cancer cells and either don't treat the mice, and here we are recording the growth of individual tumors, or we treat them with CDK4/6 inhibitor palbociclib. We are actually using a very low dose of this inhibitor because we don't want it on its own to have major function. We are testing whether it helps the immune therapy. So as you can see, there is some effect, some slowdown of the growth, but nothing impressive. And you should see here 0-13 survival, meaning all mice will die of cancer. Here, all 12 mice are die of cancer. Now, if we treat mice with the tumor-bearing mice with anti-PD-1 antibody, that consistent with what I showed you that immune therapy does work, although in a fraction of patients, in this case mice, we see not only the prolongation, but 3 out of 14 mice are cured of cancer. So cancer disappears, and they are tumor-free. But when we combine the antibody, anti-checkpoint, anti-PD-1 antibody with CDK4/6 inhibitors, then now 8 out of 12 mice become survivors, meaning we can eradicate tumors in a much higher proportion of mice, and overall the survival is extended as shown here. So here 3 were survivors, this were 8 survivors, and this is the cumulative survival curve. This is PD-1 antibody, works very well, and this is PD-1 antibody together with low levels of the inhibitor. Okay, so the summary of this last part is that cyclin D CDK4 kinase negatively regulates PD-L1 levels, that CDK4/6 inhibition elevated the levels of PD-L1 in cancer cells, and then importantly that combination therapy of CDK4/6 inhibitors together with anti-PD-1 monoclonal antibody inhibits tumor growth and enhances survival. I should add that this conclusion was reached independently by 3 groups in papers published virtually back-to-back from Jin Zhao from our institute, and also from Nathaniel Gray, also from our institute, and for Kwok Wang from New York University. And I understand that our institute will now start the clinical trials with a combination of those two therapies. I also must say that this is an equal collaboration between Wen Wei lab and our lab and Gordon Freeman lab, and those 3 individuals, Haijian from my lab, share the equal first authorship on this paper. This was acknowledgement for the second part of this talk, and this is the acknowledgement for the first part. Now, my lab is not that big. This slide is overcrowded, but this is, and actually the labor laws would not allow us to employ children in the lab. We also don't employ senior citizens, although there is no law against this, but these are just not only my lab, but also the boyfriends and girlfriends. People in my lab have many girlfriends and boyfriends and husbands and wives, and this makes it, this lab look artificially big. It's also nice because this was reunion, so there is Chuan Yen, whose work I presented to you with the crosses of cancer-prone mice. This is Yoon Choi, who carried this work forward with the conditional knockout mice. And of course, we could not have done this with really wonderful collaborators. We had the xenograft part was done together with Hui Gao from Novartis, and then with the metabolic analysis of the carbon flow, radioactive carbon flow, Brandon Nicolai from Nick Dyson from NGH helped us in those analyses. So I thank you for your attention, and if we have time, I'll be delighted to try to answer some questions.
The floor is open for discussion, so I see one question here. Arrange for a microphone.
Oh, sorry. Here.
So I'm curious about the other side of this CDK inhibitors influence with regard to the anti-tumor responses. I mean, the influence on the cytotoxic T cells, because one would be thinking that hampering with the proliferation, potential proliferation of cytotoxic T cells would not be so beneficial for the anti-tumor responses. So are you not afraid that introducing those inhibitors would actually have a problems for the immune system recognition and fight with the cancer cells?
Yeah, so that was a great question. We did not address this. So there is very important thing that I ignored in my talk, and this is the influence of CDK4/6 inhibitor on proliferation of the T cells. We are actually now trying to address this, but I should say that Jin Zhao's lab has compared the response of the cytotoxic T cells versus Tregs, CD4, CD5 positive, CD25 positive cells. And it turns out that the Treg, the regulatory T cells, are particularly sensitive to inhibition with the palbociclib. So this also helps because it reduces the number of inhibitory Tregs and affects less the CD8 positive cells. There's also another twist to this story because Nathaniel Gray and Kwok Wong has some data that actually, the former postdoc in my lab, Lars Anders, has found that CDK4, CDK6 phosphorylates NFAT, a very important transcription factor in T cells. We didn't pursue this and didn't quite understand what it means, but then Kwok took it further and showed that inhibition of CDK4, CDK6 actually causes activation of, so actually it helps activation of the immune response. But these are very early days, and we don't understand it fully, although we, and I'm sure many of our competitors, are studying it very actively.
Do you have any other questions?
So, do you think that cyclin D CDK4 could also affect abundance of other proteins besides PDL1? Because if the mechanism is via the E3 ligase phosphorylated by CDK4, you could imagine that I don't know whether any other substrates of this ligase are known, and whether they could be affected as well.
Yes. So there are other substrates of this ligase known. None of them, to my knowledge, is actually directly involved in anti-tumor immune response. But that being said, of course, through this or through other F-box protein, there could be there could be effect. So we are now trying to address it by treating cells with the CDK4/6 inhibitors and then comparing protein abundance, but not protein abundance by the candidate approach, but rather by doing quantitative mass spectrometry. So we have actually joined postdoc in the lab who is postdoc in our lab and then Steve Gigi's lab, and then we actually do exactly what you suggested. We try to look which proteins change abundance upon shock treatment of cells with CDK4/6 inhibitors.
So since breast cancer is very heterogeneous and now the biggest challenge is with triple negative breast cancer, can you comment on the CDK4, CDK6 inhibition in triple negative?
Yes.
Is it working?
Yes. But that's really a great question. That's really a great question. So indeed, there was really a seminal study of Dennis Slayman's lab. The first author is Finn. And what they did is that they took many different breast cancer cell lines corresponding to three major subtypes. The major three subtypes of breast cancers is either hormone receptor positive, so this is estrogen receptor positive and or progesterone receptor positive. The second is HER2 amplified, and the third one is triple negative. Triple negative because they don't have estrogen receptor, progesterone receptor, and do not have amplification of the HER2. So in this beautiful study, Slayman's group compared the whole panel of cell lines, and they found that the most sensitive ones, that is to say the IC50 of the palbociclib was the lowest, were estrogen receptor positive tumors with or without HER2 amplifications. These are the ones that were used first in the clinical trials. The second, less sensitive, were the tumors that amplified HER2, but they were negative for the hormone receptors. And then there was a group, it's really like the picture is striking, there are those triple negative breast cancers, which are seemingly totally resistant to the CDK4 inhibitor. Again, there is some subset, a small subset of CDK4 of cell lines that are sensitive to the inhibitors, but most of them are indeed resistant. And then the way we rationalized is that we say these tumors deleted retinoblastoma protein, because it's well established that if you delete retinoblastoma protein, you hyperactivate cyclin E CDK2, and therefore you bypass the requirement for CDK4, CDK6. The second thing is known that the tumors which amplify cyclin E through the same mechanism hyperactivate CDK2 and bypass the requirement. So the thinking was that they have other genetic reasons, but this is not true actually, because we have found, our postdoc lab, Anne Fassel, has found that there is a good subset of triple negative breast cancer cells which don't have retinoblastoma knockout loss, which don't overexpress cyclin, which have no changes in the cell cycle machinery that would explain, and yet these tumors are resistant. And we are really actively pursuing this, and I'm not going to present it yet, but if I ever get invited, like realistically speaking, in 2 years' time, then maybe I have something to show.
Thank you. Thank you very much. Peter Szczeciński, thanks for being here. I hope it was very interesting. Okay, and our second speaker in this keynote session is Dipanjan Chowdhury. He's a biochemist originally, and now he deals with all things related to DNA repair in Boston, the Dana-Farber Cancer Institute. Obviously, he might at first glance not be corresponding to the Polish scientific network's theme, but well, we got together because of the excellent and sadly missing mentoring program of the Foundation for Polish Science, and he's the guy who made me switch fields from diabetes to molecular biology and translational medicine and microRNAs and all things interesting in cancer. So, Dipanjan, the floor is yours.
Is there a slide change there?
Right, left hand.
Thank you, Wojciech. Yeah, I want to thank him and the other organizers for having me here. Obviously, I'm here because I'm Polish, but the real reason is this. So Wojciech and I have been working for several years now, and I was looking back at it, and it's quite amazing that in a short time he's one of my most prolific collaborators, and I like to believe a friend now, as we have been really, he's been visiting me in Boston quite often, so we get to spend a lot of time together, and his Skype a lot, and I'm afraid of his wife, because when I'm Skyping with him at midnight, I hate to see her in the background and imagine that I'm the one keeping him away from bed, but you know. So I'm not going to talk about any of this listed stuff. Actually, I'm not going to talk about RNA. I wanted to present a news story, something which we have just discovered and exciting, and try to take you through that. So you're going to hear a recurring theme of this. I'll try to, because Wojciech told me there's a lot of students and trainees here, so if I'm too slow, it's mainly to try to make sure everybody is on the same page with this. That's the goal. So double-strand break repair, a key question in the field is choice of pathway. So when you have a double-strand break, there are several ways you can repair. The two primary pathways is end joining, which is basically you're literally joining the ends, and there's an elaborate machinery. A factor which is going to come back a lot in the discussion is 53BP1. And the other is homologous recombination, which involves the recognition of the break with this mRNA complex, resection, which is basically chewing the ends, and then finding the homology. So pathway choice has become a very key factor in cancer therapy, and I will come to that. So you're going to hear versions of this, but I want to start by introducing it. All right, so my lab, large section of my lab now works on ovarian cancer, high-grade serous ovarian cancer. And the primary reason, I mean, we are trying to do things that are translationally or clinically important, but the primary reason for working on ovarian cancer is we see this as really a DNA repair disease, a disease of DNA repair deficiency. And I say this because 50% of ovarian cancer has a deficiency in the homologous recombination or the BRCA pathway. And as we get better at sequencing and looking at other things than just mutations, the number increases. So a large majority have a very critical DNA repair deficiency. And about 13 years ago, these drugs were discovered, and only in the last 2 years, 3 of these of the same family have got FDA approved for treatment of ovarian cancer. And my story really starts with these drugs. So this is PARP inhibitors. And what was a major discovery in our field, because it really gave the DNA repair field something specific, a drug in the field, was that PARP inhibitors specifically target BRCA deficient cells. So Alan Ashworth and Tom Halliday's lab in the UK published these seminal Nature papers in 2005, and these drugs have been in trial for many years, and again they got approved in the last few years. But what's important here is that typically if you just think of a DNA damage inhibitor of DNA repair, you would think of it as non-specific. But what was specific or special about these drugs was they were exquisitely specific for BRCA deficiency. So what is PARP? A little bit of introduction. So PARP, as its name, it's a poly-ADP ribose polymerase. Its most well-characterized role, and we are discovering new functions for this large actually class of enzymes, but is in base excision repair. So basically, when there is a base damage, and this is what is actually something happening in our body all the time. We have every second that we go with the metabolic products or replication, you have the single base damage, and you have recognition of this with a complex signaling pathway. And PARP, this polyadiporibose chain, is critical for the repair using this machinery. The BRCA1, BRCA2, as I said, this is exquisitely sensitive in these cells. What are BRCA and BRCA2 proteins? They're really large proteins. And again, as we know more about them, they were initially discovered as tumor suppressors. Actually, one of the labs in my institute, David Livingston's lab, was critical in the discovery of BRCA1. But they are actually much more than just participating in homologous recombination. They are really required for maintaining the genome. So how do they work? And this is very important. For non-DNA repair people, we very easily call them BRCA and classify them as the same. They are very distinct. They have very distinct roles. And where they are on the pathway makes a huge difference in how resistance happens or how they work. So basically, BRCA1 is really involved in the early step. I mentioned the early stages of homologous recombination. Here is a little more detail. So as you recognize the break and you chew the DNA end, resect the DNA, this is critical for the step of resecting the DNA. And this chewed DNA now goes searching for homology. And this is BRCA2 is required for that step of loading RAD51, which is coating the DNA. So this is an initiation of this pathway, and this is more in the completion or the later stages. So both BRCA1 and BRCA2 deficient cells respond exquisitely to PARP inhibitors, but the resistance mechanisms that are emerging in the clinic are going to be very distinct. And another very important tidbit, which is again very interesting, but we don't understand why. So in ovarian cancer and breast cancer, the patient population, about 10 or 20% patients have BRCA1, BRCA2 mutations, of which it's largely BRCA1. So BRCA2 is less than 5%. But now there is very exciting data that in prostate cancer and also in pancreatic cancer, significant number of patients have mutations in BRCA, and it's the reverse. There the majority is BRCA2. We don't understand why. PARP inhibitors work for both, but again, resistance will be different. So how do they work? So why are BRCA deficient cells specifically sensitive to PARP inhibitors? So the first mechanism that has emerged, and we kind of people agree on it, is the concept of synthetic lethality. People who work in yeast or in model organisms have used this for many decades. It's a little more difficult to do with mammalian cells. Now with CRISPR technology we do it. But the concept is that if you have deficiency in a pathway, you might be over-reliant on another pathway. So if you can target that other pathway, you can kill the cell. So what happens here is, as I said, typically PARP is involved in the repair of a single-strand break. If it's not repaired because you have the PARP inhibitor, then the single-strand break gets converted to a double-strand break during replication, the phase where you need the homology-directed repair, the BRCA pathway. Now you have a defect here, this break doesn't get repaired, and you induce apoptosis. So basically, the lack of single-strand break in a normal cell, if you have normal HR, then this will get repaired. So that makes it specific. The other mechanism which has been, I would say, discovered in the last 5 years more recently with all the clinical trials is something called PARP trapping. Basically, the idea is that the PARP inhibitor is not actually inhibiting the catalytic activity of the enzyme, which we always think intuitively an inhibitor does, but what it is doing is it's trapping the enzyme at the lesion. So when the lesion, as I said, recognizes the break, the single-strand break, you trap this big enzyme there, keep it there, and that in itself is enough to trigger the cell death pathway. We don't completely understand how this happens, but you can imagine that an enzyme trapped, particularly when it needs to, it's a very, very fast process, you're really messing up a signaling chain, and that's triggering death. So it was all very good in terms of, I told you that they work exquisitely in terms of targeting BRCA cells, but all of that was largely from preclinical studies. As this has gone to the clinic, what is becoming increasingly evident, and it's not a surprise for any cancer drug, is there is a lot of resistance. And what was surprising, I think, to the physician population is that actually a relatively small fraction of the BRCA tumors are responding initially to PARP. There is 30% is a large number, considering there are no drugs. There have been no drugs in ovarian cancer in the last 25 years, new drugs. So this is a new drug and it works, but this number potentially should be increased. So what are the mechanisms of PARP inhibitor resistance? The mechanisms that have emerged in the clinic that we know are happening in patients, very simple. You have a mutant BRCA1 protein. You basically have a somatic reversion. The tumor selects out something where this mutation is gone. There's a frame shift and you lose the mutation and you restore functional protein. Another mechanism is there are several tumors where there is no mutation in the protein, but you suppress BRCA expression by methylating the promoter. So now you lose this epigenetic regulation and you restore normal expression. So basically the selection of the PARP inhibitors or selection of death is causing this. Another one is again very similar, where there are two alleles, you splice up, there's a hypomorphic, and you restore the protein. So these mechanisms are relatively the same idea. You're getting back wild type protein and getting back HR function. This is a very, very common theme with tumors. Imagine the complexity. You cause the tumor by having a mutation, but then to maintain the tumor for its survival, for its health, you basically restore the tumor to a normal DNA repair machinery. There are several other mechanisms which have emerged in model systems. So how many of these are actually happening in patients is really not clear at this stage. One is loss of PARP expression. This actually connected to the PARP trapping concept, that obviously if you don't have the PARP1, the main enzyme, the inhibitor is not working. But what's clear is then in that case, you don't have this trap situation, so you get resistance. This is something I alluded to in the beginning, and we'll come a lot to this. This is the interplay, the choice of break pathways. 53BP1 is the guy who's taking the double-strand break to the end-joining pathway. So who gets to the break first? So a lot of the resistance mechanism is coming where normally if end-joining happens, you cause mutations, and there's enough mutations, the tumor dies. Here, without end-joining in a tumor, you're restoring the BRCA1 pathway. This is specifically happening only in BRCA1. Again, we'll discuss this in detail. Another pathway which is relatively understudied is translesion synthesis, TLS, the polymerases. Basically, very simply speaking, you circumvent the break. You don't deal with the break. You skip the break using a specialized group of polymerase, and you continue. So CHD4 was shown to be important in that. So if you don't have CHD4, you actually can do TLS. So now you're not dealing with it. You're going and repairing the break. And the other, this is probably the most common drug-resistant mechanism that's been studied for a lot of drugs. Basically, your pumps are altered, where now you are just pumping the PARP inhibitor out of the cell, hence your cell is not responsive. So what we set out to do is all these different mechanisms that have been found, some in the clinic, some in model system, have used candidate-based approach. The idea is, can we systematically map out every possible way, at least within an experimental system, that a cell, a BRCA deficient cell, can become PARP resistant. So for that, we use the CRISPR screening technology. Again, I imagine most of you have been exposed to the Cas9 CRISPR system. It's a new gene editing tool, not that new anymore, but it's really changed how we do genetics in mammalian cells. This allows us to knock out genes with relatively high efficiency, much more efficiently than knockdown. The knockdown with siRNAs was how these screens used to be done. So in our case, in collaboration with folks at the Broad Institute in Boston, we could knock out 16,800 genes in 3 patient-derived BRCA1 mutant cells. So again, this resistant mechanism would be specific to BRCA1, and we can discuss this in detail later. And the idea is that if we knock out all the genes in these cells and try to find the loss of which gene causes survival. So you're selecting out for things that survive the drug treatment. And we did it for both cisplatin and PARP inhibitors. And the reason to do cisplatin, which is supremely important, is today, everywhere in the world when there's an ovarian cancer patient comes to the clinic, they all get cisplatin-based therapy. That's the only therapeutic modality. PARP inhibitor is new. So there is a lot of pre-existing data about response. And cisplatin therapy also goes through the same sort of HR pathway. BRCA deficient cells are responsive to it. So we wanted to compare both. So the system here is simple. We have cells which are stably expressing the Cas9, the endonuclease that cuts. The guide RNase, which gives the specificity for each gene, is expressed as a lentiviral library. And then you select, look at these cells, drug selection, you have survival, you sequence. The data sort of looks like this. You get enrichment of guides, at least few guides, 2 guides per gene. We have about 6 guides per gene in this, actually 4 guides per gene in this particular library. And this is a clinical PARP inhibitor, olaparib, and then cisplatin. What's interesting about, I have never done an siRNA screen, but I would always wonder when these really high-profile papers would come out, that how did they pick the 53rd hit and knew that was going to be important? So when we got our list, we decided to pick the number 1, which makes sense. What's interesting about CRISPR is it really validates. So again, with siRNA screens, when people show the validation, you see a large majority are off target. That is very rare in a CRISPR screen. What's exciting here is I'm going to tell you a story about DNL1 and ATMN. Basically, these are connected. And we really picked DNL1 because it was our number 1 hit. But these 3, so 53BP1 we already knew was number 3. These 2 proteins, there are 3 Nature papers, 1 Cell paper, 1 Nature Cell Biology paper, and I'm missing a few papers coming out on how these 2 proteins cause PARP resistance. They're all coming out or have come out in the last few weeks. So it's incredible that in a screen completely done differently with different cell lines, you basically get everything that's expected in your top hits. So it gives you assurance that you are actually looking at things that make sense. So we picked DNL1 largely because it was number 1, and ATMN because it works with DNL1. It was again on the top hits. So what was known about these 2 proteins? So ATMN interacts with ATM. It's a ATM interacting protein. For people in the DNA repair field, ATM is one of the most important signaling molecules. It's a PI3-like kinase. What we know from the literature is these guys had very little to do with double-strand break repair. So if you take these out, there's not much of an impact on DSB repair. They've been shown to have an impact, ironically, in BER. ATMN is actually a transcription factor, so it promotes DYNL1 transcription. You'll see some results on that. And they have been really implicated in oxidative stress. Not a whole lot, sort of an obscure bunch of papers. So when we took our knockout clones, one thing is striking is if you knock out ATMN, you lose DNL1. If you knock out DNL1, ATMN levels are unchanged. This is the validation of the screen. So we make different clones. ATMN knockout, DNL1 knockout, 2 different clinical PARP inhibitors, cisplatin and carboplatin, cisplatin slots. you can see whatever you do in these cases, you get very high resistance to these drugs. Then even with CRISPR, and this screen was done about 4 years ago, 3 or 4 years ago. So we start rescuing just to make sure even with the guides there is no off target. And this is DNL1 knockout cells where we are rescuing with DNL1 constructs. But what was striking is that if you take Atman knockout cells, we can rescue the effect with DNL1 construct. So this really made the relationship epistatic. It made sense with what I showed. Basically, if you knock out Atman, you lose this. But if you put this back, then the phenotype is restored. So that's basically the bottom line. So Atman works through DNL1 to cause sensitivity. What is DNL1? So this is where it's confusing. It's confusing for, I believe, the field. So this is DNL1, dynein light chain-like protein. Dynein, as we know, is cytoskeletal and all of that. LC8, light chain 8, it's extremely small. It's an 8 kilodalton protein. It's also called PIN. Now the confusing part is if you do a literature search with DNL1, you get a set of papers. You do LC8, you get another set of papers. And we found it to be PIN the last, you get very different papers. And it's very important because this is actually an extremely well-studied protein historically, and it binds several complexes and disrupts them. So this was something which is important to know. It has nothing to do with its function as a dynein light chain-like protein. So another very critical reason why we followed this up, and this is something we do in the lab routinely, and this is I would say what I have been educated being at Dana-Farber is we can study whatever we want, but if it's clinically relevant, it's even better. And we can easily, particularly if you do screens, you can really choose something that has a clear clinical relevance from all the publicly available data. So we use this data a lot, and I'll give you several examples. So this is very standard. This is the Cancer Genome Atlas. And what we look here is for progression-free survival. And as I said, one thing with ovarian cancer is every patient gets treated with platinum. So this is pre-treatment data analysis of Atman or whatever protein you are interested in. And what this shows you is that if you look at the BRCA mutant ovarian cancer patients, again, all of this is publicly available, you can see that high level of Atman correlates with poor survival. All these patients have got treated with platinum. So this is really a marker for resistance. Progression-free survival can be equated to resistance. So it already shows you that high levels of this protein, the other way around, low levels of this protein correlates with resistance, exactly what we got in our screen. And this is specific to BRCA mutants. If you look at the BRCA wild type, there is no effect. We also looked at BRCA2, there is no effect, and it makes sense. Going back to this pathway. Again, here we are, a double-strand break. It's BRCA1 deficient. We have a factor whose loss is somehow restoring PARP inhibitor sensitivity, which is specific to this pathway. So one of the markers and a factor in this pathway is RAD51. So as I told you, there are several mechanisms by which PARP resistance happens. So we wanted to know whether now in a BRCA1 deficient pathway, have we restored this pathway when you take DNL1? And to do that, the first assay we do is look at RAD51 foci. So if you look at RAD51 foci, you'll see exactly what we sort of anticipated, that in the control, you have much less foci. Remember, this is BRCA1 mutant, so it's never not there. There is some RAD51 foci, but you significantly, dramatically increase the number of foci when you take out DNL1. So we have now restored the pathway. Now the question is, as I said, BRCA1 works upstream, the initiation step. So this is the key step, where you resect with BRCA1, you don't have BRCA1, you are not resecting, and you are going this way. And there has been a large body of literature, really very elegant papers, showing how 53BP1 plays a role as a counterpart. So what happens is a 53BP1 mediated repair, no resection here, actually eventually kills the cells because it's mutagenic, and there is enough mutations the cells die. If you take out 53BP1, you restore this. So it appears that the only job, in this case BRCA, one of the primary jobs BRCA1 was playing is keeping this guy away. So people have shown if you take out 53BP1 BRCA1, you restore end resection. So then it turns out DNL1 is known to interact with 53BP1, and they map the domain, so it's well-established interactor. So we said, okay, simple hypothesis is DNL1 interacts with 53BP1. If you lose DNL1, you lose this protein, and that's why you lose the function of this protein, and that's probably the mechanism. Well, not true. So if you take out DNL1 and look at 53BP1 foci, although there is a mild difference, it's not significant. I showed you the resistance. The resistance is really dramatic. So this is clearly not dramatic. So the 53BP1 foci does not explain this phenotype. So then we started assaying for resection. And the assays for resection are RPA foci. So if you have resection, you have single-strand DNA, and you have coating with RPA. So one of the assays is RPA foci and RPA phosphorylation. So we can see that we see increased RPA foci. So it makes sense. You have restored RAD51. You are basically showing that you have restored BRCA1 function here because you are seeing resection. Increased RPA phosphorylation, again a marker for resection. But resection is actually a relatively controversial step in the sense of people disagree on which assay is a true assay for end resection. So this is more, I would say, the aficionados in the double-strand break repair field. So we really wanted to make sure that we are indeed studying end resection, and there are some cool new approaches. So one of the approaches we adopt is a site-specific break system. So this is a system where we can make 80-odd breaks in a genome, and we know every break that is there. And then you can look at as many as you want, 3, 4, 5, and look at resection at these breaks. You can do it in a genomic way, which is expensive, or by PCR, where you can look at the breaks. And what's nice is you can look at how far the resection goes. So you're making a break here, you can follow the break and look at resection, the amount of resection. So it's a very nice system to study this and quantitate this. So basically you measure end resection using the system, and you can see that if you knock down BRCA1, you decrease resection, both close to the break, further out from the break, and now if you take out DNL1 and BRCA1, you restore this back. Another independent system to say that indeed we are bringing back resection by taking out this factor. Yet another assay to look at resection, and I know this might seem extreme, but trust me, when we send the paper out like this, you have to satisfy every reviewer's favorite assay. So this is somebody else's favorite assay, where we look at resection at a single cell level, looking at DNA fibers. So these are basically DNA fibers where we are actually looking at single-strand DNA, and we can measure the amount of single-strand DNA and say that we have resected the DNA. Okay, so back to this model. So what we have so far is, we have a system in this choice. Now DNL1 is negatively regulating end resection. So if you don't have BRCA1 and you don't have DNL1, now you restore end resection. So this is a negative regulator which is keeping back resection. Particularly you see this when there is no BRCA1 and you're restoring it back. So this is again where we go back to clinical data, again publicly available clinical data, and it's actually amazing the power, and I really say this for students and trainees who are learning how to analyze genomic data and are learning computational tools. It's amazing what we can do sitting anywhere in the world. So this was a paper published a few years ago from David Botel's lab, and honestly I can tell you, I can admit that when I saw this paper, this was a Nature article, I mean I had 3 questions. How much money did it cost? Why is it a Nature paper, and how boring? Because all they did was they did whole genome sequencing and transcriptomics and microRNA sequencing as all the omics that you can think of in 92 patients, 114 tumors. There was a wealth of data in it. So the way we analyze this data, and when I say we, it was done in collaboration with my friend Shubha, who's a computational biologist, was look at these things called somatic structural variants. Somatic structural variants, because they are sequencing both the germline and the tumors, you can think of this, or I think of this, as DNA lesions and repair. So there are variants, there's mutations that have happened, or changes that have happened. So imagine this. Basically, we are analyzing 36,000 breaks and repairs and seeing whether our proteins of interest impact these or not. Okay? And it gets better. So as you would expect, so if you look at all somatic structural variants, or you can look at the things that make more sense to you, like for us, we look at deletions, duplications, insertions, translocations. So these are the ones here, intrachromosomal changes. As you would expect, and you classify these tumors based on BRCA1 expression. Transcriptomics data was there. So if you look at the BRCA1 expression, as you would expect, that the low BRCA1 population, you have increased structural variants. Makes sense, right? If you take out a very important DNA repair protein, you have more mutations or more changes in the genome. Now, if you take both out, if you look at the tumors which have low DNL1 and low BRCA1, you basically bring it down to the baseline, to the line that was DNL1 alone. So this really tells you that in the absence of both proteins, you're restoring some form of DNA repair, that you are changing across such a large number of structural variants, you're really reducing the number. Okay, even better. So we have the sequence of each variant, each structural variant. So you can look at homology around them. So the algorithm allows you to look at homology around these structural variants. And the advantage of that is if you're indeed using homologous recombination, BRCA1, you can see increased homology above 10 base pairs around them. And if it's not, it should be different. So the data is clear. When you have low BRCA1, homology goes down around the structural variants, very significant. If you have low BRCA1 and low DNL1, you're back to baseline. And even more satisfying, if you have more DNL1, which we can't really recapitulate in the lab setting, you seem to have more homology. I don't want to talk about this because we honestly don't understand this, but this is clear. That if you don't have BRCA1, you're decreasing homology, so whatever your variation is, your variant is likely being repaired by other pathways. Now, when you take out both, you're restoring it. This also suggests that potentially, if this is happening in a tumor, these things are happening at the same time. Because if indeed BRCA1 was lost and it went through several generations or proliferations, then the scar would have been where there would be no homology. So very likely these are getting suppressed around the same time. All right. So now the mechanism. So what I've established so far is that we have low BRCA1, that low DNL1 is indeed restoring resection and at the breaks. How do they actually function? So this is where we started digging into, and it's kind of unfortunate that we found a lot of very important papers which we had missed early. One of them was a paper from Solomon Snyder's lab, a 1996 science paper, where they showed that this pin, they called it pin, actually breaks dimers. It's a multimerization hub. It's an 8 kD protein which breaks or makes a lot of protein oligomers or dimers. And for example, it breaks NOS, it breaks BIM, so it acts as inhibitors of these. And the structure of this protein has been solved. So it functions as a dimer in some cases, and the dimerization is dependent a lot on the serine 88 phosphorylation, but there are some other critical residues which is formed. It can also function as a monomer, which we find. So then we collaborated with Jean-Yves Masson, who is a lab in Quebec, an excellent biochemist, real biochemist, where they reconstitute the resection system. So in this case, they have reconstituted all these factors which are required for the early step of resection, and they have a clear resection assay. You can see the resection can be assayed. And it's a very simple question. If we add our protein into this, does it block resection? And if we have mutations that don't, what can we learn from that? So we did that experiment, and as you can see, if you add the wild type, it's actually quite nice. You can clearly block resection. This is the purified proteins. And then we found 2 mutants, and thankfully actually 3. One is not shown. The C2A, which doesn't block resection, and the phosphomimetic, which blocks resection like the wild type. So this system now lets us really go into the depth and say, how is this affecting resection? So we made the mutations that I just showed you, and then we started looking at interaction. So all these proteins were in the mix, so we started looking at the interaction in cells, and we find that the wild type interacts with all of these. All right, but this is still, one could argue at this stage that we have in here an in vitro system where it's purified proteins. Here here it's cell lysate, so the compartments are all broken. So do they actually interact in cells? So we did the experiment. This is actually a cool system that we use where we can tether our protein of interest in the cell and then look at interaction with our any other protein. So DNL1 has been tethered. It's a nice red blob. Now, when we express all these other proteins, you can see the colocalization. So they're indeed interacting within the cell. Going back to these mutants, we did the interaction with the mutants. As we saw in the in vitro system, the D mutant interacts really strongly, wild type interacts, but these two mutants which did not inhibit resection in vitro don't interact in cells. So basically the interaction seems to be key to the inhibition of resection. That's fine, but does that impact the ultimate assay? This started with PARP resistance. Is the interaction really involved in PARP resistance? So we can do that too. So we can take a DNL1 knockout cell, put the mutants back, and as you can see, the wild type and the phosphomimetic, which interact, actually sensitize the cell, so they rescue, as you would expect. These two guys don't interact, and they don't rescue. So this really tells you that the interaction is critical for this phenotype. And the final piece of data, which is going back to the in vitro system. So as I told you, we found that all these mutants, these two mutants don't inhibit this, and the wild type inhibits. So using this, we could see what is it the factor. There are 5 factors here. Which is the factor that it really interacts with in vitro? And the answer is MRE11. MRE11, which is probably the most important factor in this case because it's the part of the complex that recognizes the double-strand break and has endonuclease activity. We can see that in vitro, DNL1 and the phosphomimetic, which both inhibit, interacts with MRE11. So I'll stop and give you the very relatively simple model we have, which is in a BRCA deficient cell, you have Atman, which induces DNL1, inhibiting MRE11, that leads to the loss of end resection. Now, when you lose these proteins, and this is happening in a patient, as I showed you with TCGA, you are now increasing MRE11 activity, allowing end resection. I'll acknowledge the folks who helped us and did the work. So this is really led by Ijo. He got a lot of help from other lab members, but this is really his project. We have an ongoing collaboration with Alan DeAndrea and Panos, who's an ovarian cancer medical oncologist. I learn a lot from him. And Jaiv and Shubho have mentioned the data that we got and the funding. Thank you.
Right, so this is the method going slow on DNA repair for you. This is slow and easy, accessible piece of data. But if you have any questions regarding the talk and specifics, feel free to ask them.
Please.
Excellent results. I would like you to comment on the non-homologous enjoining in this situation when both BRCA1 and then LL1 is lost, because you said the homologous recombination is restored, but maybe this is the other part also more active.
So, you know, we don't see any difference. So I can tell you what's interesting. This is a very important area for us. So some of the other factors that have been discovered by the screen, the ones I mentioned, FAM35A, they're called in the Shieldin complex. They are actually now shown to be NHEJ proteins. What's interesting is if you knock out DNL1 in a BRCA proficient cell, which is where we have been looking, there is no impact on NHEJ. The cells are not, there is no impact on radiation sensitivity. We have done class switching, so there is no effect. I can tell you we think it has a role in replication fork stability, so the other area, that's probably where it's keeping MRE11 away. Its physiological role in DNA repair, but nothing in non-homozygote joining. So.
Any other questions? It wasn't arranged prior, so.
No, I have to come to Poland to see Peter.
That's right. The question: how much of this is acquired resistance, and how much of this is pre-existing resistance?
So the TCGA data is all pre-existing because it's pre. But you know, we don't have access. We will eventually have access to PARP patients who are now becoming resistant, and pre and post. So we will investigate. Right now, all the data we have is pre. But you know, it's not inconceivable that you can acquire resistance by knocking this out.
Right. And then you did the loss of function screen with the CRISPR. Did you
Yes.
try also gain of function screen to see overexpression of which?
No, we haven't done gain of function. Actually, it's quite, this protein is essential. It's an essential protein. We cannot, overexpressing it in cells is, you have to not have P53 and all that stuff. So it's quite, because it has so many other roles, but we have not done a gain of function screen. We have only done a loss of function screen.
I'm curious whether this mechanism, it only operates in the condition of PARP resistance, or there is a physiological role for this. I mean, I'm thinking like nicotinamide, as I can understand.
So it has
that's
a resistance in platinum too. So it's for everything. So what is interesting is PARP is specifically is a new drug it's giving, but cisplatin is given to lung cancer, testicular cancer, cervical cancer. There's a lot of situations. So we want to look there, but the physiological role, and I was alluding to that before, we think the physiological role is in replication fork stability. So there's a large body of emerging literature in the genome stability field where in a regular cell, there are lots of difficult to replicate regions and the replication fork gets stalled. And that's where the role of MRE11, the endonuclease, becomes important. But what we think is, it's like having anything. This is an endonuclease. You need to restrict its activity. So if you don't have an inhibitor like this, then it destabilizes. So we have data that in a normal BRCA proficient cell, if you take this out and you look at replication forks, you actually see a problem. You see less rate of replication and you see stalled forks. So that's basically where...
In the absence of
DNL1, yes.
Okay.
And we are actually doing electron microscopy to visualize these to really know what happens. Is it fork breakdown or regression? So that's probably in the DNA repair area, I think that's where its physiological role is.
We have time for one last question.
Yeah, I'm sorry for running a bit over time, but I really wanted to make it slow.
Okay, so my question is about this example which you showed that it is possible to have a somatic reversion. So you have, let's say, mutation in BRCA1, and next the BRCA1 behave like a wild type.
Yeah.
So this could be, maybe this is in the complex with molecular chaperones like HSP90, which have such a buffer effect. This is one thing, and another one, maybe you have an extragenic suppressor, which actually suppressed the first mutation in a case of.
So you're suggesting that DNL1 is allowing BRCA1 reversion? Right, so we've tested that. So we can, because we found this in patients, so we then took BRCA1 knockout cells and we have generated BRCA1 knockout cells to make sure that BRCA1 protein is not coming back. So that, the second question I didn't get. So what was your second question?
Well, it is possible that during the carcinogenesis you are inducing another type of mutation which suppress the first
for sure.
mutation.
I mean, in the patient tumor, for sure. But that's the beauty of the in vitro system, is you can go back and literally isolate everything and show at least the phenotype we think is happening. So, but in a tumor, yes, sure.
Thank you very much.
Thank you.
Talk. And now you are faced with a very, very difficult choice, because we will have the free concurrent sessions. If you fancy a bit of bioinformatics and big data analysis in the afternoon, please go to room 117.